Okay, this recording is for Monday, come on, wake up. Monday the 9th. Okay, and let's just make sure this share is working. Good. All right, Monday the 9th for week 2 of LBT 280. So it's being recorded. That's not what I care about. So what I'm going to do is, first off, go back to the modules here and just go through what we're going to be doing this week. I'm going to do some recording on collection, processing, and preservation of urinalysis samples, and then analyzing those samples. We were going to do microscopic urinalysis tonight, but we'll do that on Wednesday. We will instead get started on the diabetes insipidus assignment. And there are 2 forms of diabetes that are going to be discussed. One is central or neurogenic diabetes, and the other is nephrogenic or generated by comes out of the kidney. And the central just comes out of the central nervous system. So what you're going to be doing is actually splitting this up. I'm going to give, since Maddie is the one here tonight, I'm going to let her choose which one she wants. Neurogenic. Okay, so Brandy, I want you to do nephrogenic, and Ariel, if you end up seeing this and are trying to catch up, choose one. Do all the research. What is the disease? Rate of occurrence, meaning what percentage or what number of people out of, say, 100,000 are likely to have this disease? What's the cause? Because they do have different causes. And what treatments are available? So references, since each of you is choosing one of these topics and writing a report. A presentation. The presentation can be a PowerPoint, which I would like to see done on Wednesday. So the presentation can be a PowerPoint. Okay, short, maybe 10 slides-ish. 10 to 12 is probably a good number to cover all of these things. And then plus a cover slide and a reference slide. So maybe 12, 14 slides, 14 or so slides in this. Your references, you'll need at least a couple of references. So do your research. And after we get done with these lectures, I'm going to stop the lecture part of the class and let people get started on the assignment. So that is what we'll do on Wednesday. We'll do those. The discussion this week is about the medical assistant's role in urinalysis, processing, testing of urine and stool specimens. So here is a listing of some of the reasons doctors may order a urinalysis. So look at each of these and the different things that you have to do and the different advice you have to give to the patient. Because as you see from the lecture on collecting and preserving specimens, different purposes require different kinds of collection strategies. So that's going to be your discussion. I've already made one post, so you can read that and get started. There is an assignment this week, which is to look at the role, is to go on to your Medline University account and take the course that's listed there, Nutrition's Role in Urinary Tract Infections and Hydration. If you don't have a Medline University account, just go to Medline University. Just type that into your browser and set up an account. These are free. They're excellent tools. And when you're out there working, they will be great ways to do continuing education. So get used to that. Medline is a major supplier of supplies to hospitals, doctors' offices, and places like College America. So they are trying to do their best here with Medline University to give back to their community. Okay, so there at the end there will be a quiz, and you'll have a final score. That's what I want to see, the final score, not the certificate of completion. If you want to submit that too, that's fine. I would tell you right now, print it out and keep it for your portfolio to show that you have done this. Okay?
Does the screenshot count? If you don't print it out, we can make a folder.
If you make a folder and just download your certificate, yeah, you don't need to give that to me. You don't need to take a screenshot of it. But I don't want you to make a mistake and take a screenshot of that and not your final score, because I don't think you can go back and get it. Does that make sense?
Well, couldn't you redo it? If you had to?
I don't know. I've never even tried to redo. I'd have to look. So I've never, but so basically you're going to be spending some time on that. So this will be part of your hybrid part of the class. So go ahead and do those as well. You can do those. You don't need the rest of these lectures to do the nutrition role in UTI and hydration. Nutrition is, we all know, an important part of health. So urinary tract infections and in hydration, yeah. Okay. Yeah, the course name is Nutrition's Role in Urinary Tract Infections and Hydration. So if you just click on that or find it, you should be able to do that. Okay. So, and then there is obviously an assessment for the week. We'll have the in-class activity. We also will have a midterm exam. So the assessment will cover variety of things that are this week's topics. The midterm will cover the first couple of weeks of class. So then you have some videos to watch. The hybrid video on interpretation of urinalysis is about 15 minutes, and it's pretty good. And the videos on male reproduction system are also pretty good. So
I'm gonna do all this by
Saturday.
Okay.
That's the week's work.
Well, I'm just sitting here thinking to try and do the videos and do the assignment.
The videos are relatively short. Okay.
Oh, but I get time here to do the assignment.
Yeah. We'll have time in class tonight. We're going to go through the lectures and then we'll get started on these things. So you can do these assignments in any order you want. I would.
Well, I need my one assignment with the PowerPoint.
Yeah, that should be your first priority. And you need to do some research. You need to search out some things. So it shouldn't, and you can get started. But the videos, these videos, each of these videos is relatively short. And then you have until Saturday night to finish everything up. Okay. All right. So let's go to the collection, processing, and preservation PowerPoint. Okay. So I'm going to do a slideshow here. Is that coming up? Yeah, there we go. Okay. So the 3 steps, collection, processing, and preservation. Well, obviously, collection has to be number 1.
Right.
Preservation may be necessary before processing, before doing the testing, the analysis. But generally speaking, these are the orders in which it's going to be done.
What would it mean to be sitting there for something to accumulate, or?
Yeah, when you take a sample, it can stay out at room temperature, covered obviously, for about 20 minutes. If you cannot process or test it in that 20 minutes, it should be refrigerated.
Okay.
And then you bring it out, let it warm up to room temperature, and go through the testing. Processing means often transferring it, which we'll go into, transferring it to a sterile tube for culture. If you're worried about UTI, you may want to culture whatever's there. In fact, you should, but quite often it's not done.
They just give you medicine.
Yeah, they just give you medicine. It takes 3 days to culture. And sometimes much longer, depending on the type of organism. It can take up to 2 weeks to do a culture. And you don't want someone suffering for 2 weeks.
Right.
So we'll make our best guess. The vast majority of UTIs are really a transfer of organisms into the body from our skin, which means they're mostly going to be streptococcus of some sort or staphylococcus of some sort, because we all carry that.
Mm-hmm.
But it could be a number of others. It could be Proteus species. It could be Klebsiellas. It could be a variety of things. But a lot of times we'll just start getting an antibiotic preferably while they're identifying the bug. And hopefully that'll help. If it doesn't, then you need a new antibiotic. And that's really why you're almost always told if you don't feel better within a couple or 3 days, come back. Yeah. It means they've given you the wrong medication. So we have a few cartoons in here. This one is from the viewpoint of the elephant calf, wondering what that guy is doing with putting a cup underneath. So a method of collecting a specimen, especially for something as large as an elephant, is to just try to get lucky and catch them when they're urinating. That doesn't always work. One time years ago, when we had a little West Highland Terrier, that was our pet, he had some sort of suspected infection. We were told to get a urine sample from him. And we said, how are we supposed to do that? We'll just follow him around, and when he lifts his leg, catch it. Well, we did. My wife was there, and he'd stop, lift his leg, and she'd shove the cup under, and he'd do one of these and jump aside and go walking on and not do anything. So we finally got him. He finally just had to let go. So my wife caught a sample, and then the dog suddenly dropped his leg and knocked it right out of her hand. So we finally got a sample, and then the blasted vet, we walked in with it, and he says, "Oh, well, I'm just going to catheterize the dog." We said, "Thank you. Thank you very much for putting us through all that nonsense." But that's what happened. So types of specimens. We have different types of specimens for different types of purposes. We have specimens that we want to catch every drop of urine for 24 hours from the patient. Because the composition changes at various times of day. But we also have other specimens. One is a first morning specimen. Basically, as soon as you get out of bed, go in and pee in a cup. First morning specimens are preferable for things like pregnancy tests.
That's how I did it.
That's how you did it, yeah.
Because I peed on it at nighttime, then I peed on it in the morning, and then I was pregnant.
Yeah, because the hormone, the human gonadotropic hormone, the chorionic gonadotropin, concentrates overnight in a bladder that's not being regularly emptied and refilled. So that's why what we do, the reason we do that is to get, you don't normally generate HCG. So, but when you do, it's in small amounts, even when you're pregnant. So we want to have the biggest possible sample. There are other things that we do that for as well. We have the random single test any time of the day. You go into the doctor's office and they say, "Here, give us a sample." They haven't asked if you've eaten, you haven't done anything else. We've got timed specimens. It can be 4, 8, or 24 hours. We can catheterize and get the contents of the bladder. And then finally, we have the clean catch specimen, which is a specimen that we should be doing for most of our tests, other than the timed, up to 24 hours when we want every drop. Because what we want to do is void the first couple of ounces of urine and then collect after that. And then when the cup is full, just get rid of the rest. That clean catch allows any sort of bacteria or anything else that happens to be resident in the ureter from migration through the opening gets washed out, basically. So we have a better chance of getting a clean sample. Remember that urine should be sterile. That first ounce or so may not be sterile, but that might have nothing to do with your urinary system. It just might be because bugs migrate. And most of the time we just pee them out and they don't cause a problem. So the first morning specimen is the best for a lot of tests. And it allows us to see abnormal levels of cells and chemicals and all sorts of other things that might be confusing to the test or might be there that we need to see. It also avoids activity. Activity actually causes us to generate things and get cellular debris and other things into our urine during the day. And of course, diet. The contents of our urine are going to be really based on what we eat. Okay? Or we ate a lot of sugar.
Right. So we're eating for eat a bunch of carnival cake and stuff like that day. So then you have all this sugar in your system. And it could make it look like you have like pre-diabetes or diabetes even.
Yep.
Even though it's just you just sugared yourself up.
That's exactly
and
right.
The next day, you're perfectly fine.
Yeah. So that's one of the things that we like about the first morning specimen. If you're told to get a first morning specimen, and it's going to be more than about an hour before you can get it analyzed, which in most cases I would bet is going to be the case, you should cap it and refrigerate it. And remember that urine is pretty safe. You don't have to go, ew, urine in the refrigerator. Don't have to worry about it. So another little joke. Tapirs, which are a member of the elephant family, can spray 7 to 10 feet backwards. So, and they like to do that. That's a way to chase off humans. Humans are their enemy. So random samples are just the most common, the ones we can get. They're used for most of our screens like we did in class last Wednesday, our general UA screens. We also use them for random samples for drug testing because we don't want, the reason we do random drug tests is because we don't want people abstaining and changing the results. We want to catch them. And, you know,
Not only catch them, but if you don't know what somebody's taking.
Yep.
Off of the street, like in street drugs, or whether it's even some, what is it called? Herbology.
Yeah,
Medication.
a naturopathic or herbal med, yeah.
And they don't want you to know about it. But we need to know, especially if we're trying to diagnose them with a problem, when these certain things could be causing the problem, and if they're not in the analysis and in the picture,
Yep,
then we're missing a piece, and we can't get the correct diagnosis.
that's true. That's true. So these samples might be just ordered throughout the day at random times to check for things like maybe blood in the urine, which can appear after hard exercise. And so, you know, we want to know if this is a one-time thing or if it's a continual problem. If we have abdominal pain that we can't localize, say, to an appendix, then we want to know what's going on. That can be an indicator of a UTI. Or if someone is having a burning sensation during urination, that could be an indicator of a UTI. It could also be an indicator of changes in things like pH of the urine. So, you know, we should have these done fairly regularly as a part of our routine check on our overall health. But the best sample is still going to be something that's been in the bladder for at least 2 to 3 hours. And the reason for that is that the specimen is concentrated enough to be fairly accurate, not perfect necessarily, but we really want to get as accurate as possible. And if we're trying to determine what's going on, we have to have a chance of seeing it. Most of our just quick CLIA wave tests aren't real sensitive, so we need as much as possible. So, and we also need as much as possible to be able to dip the dipstick in. We saw that last week, yeah. So these can be collected at home or at the office. Now, some tests you don't want collected at home. If you're doing drug screening, you don't have it collected at home. You don't know whose urine that is. Yeah, and we really, ideally, the patient should use at least a wet towel or wet, paper towel or cleaner to wipe the genital area. So, pardon? Baby wipes, I don't recommend those, especially on genitals, because genital tissue is real sensitive, and baby wipes have alcohol in them.
But we wipe our babies with them.
We wipe their bottoms, not their genitals.
We wipe the outside.
Yeah, we wipe the labia, yeah, the outside of the labia, but and the penis on the boys.
Yeah.
But you know, that would be if it's acceptable to you, that would work. But generally speaking, just a clean, wet paper towel
Okay.
will work just fine. But we want to clean that. The They're suggesting moist towelettes with a cleaning agent. That depends on the patient. I've people who are fairly clean, you can clean with a wet towel.
Right.
People who, I mean, if somebody just came off running a marathon, I'd say, well, you want to lose something a little more. Of course, then you're going to see blood in their urine too because of their exercise, and protein because of all that muscle protein breakdown products. So, but again, we want to have about, and you know, soapy water works pretty well as well. We want to collect about 10 milliliters at least. 10 milliliters is about a third of a fluid ounce. Yeah, about a third, no, about 2/3 of a fluid ounce. Yeah, 3
So 10.
tablespoons to it. Yeah, a third of a fluid ounce.
How many milliliters again?
10,
10 milliliters. Okay.
2 teaspoons. Yeah, and there are 6 teaspoons to a fluid ounce.
Okay.
So yeah, you want at least that. Most of the time, you're going to get essentially a full cup because most of the time people have plenty. So the procedure they discuss here, again, you should instruct, and this is for you talking to your female patients. Thoroughly wash your hands and dry them, and then with one hand expose the genital tissue, and with the other do the washing, and then wipe always from front to back because you want to wipe toward the anus because you don't want to wipe fecal matter from the anus into the genital area. That's a great way to contaminate your specimen, but also a great way to give yourself a urinary tract infection. In fact, it's why women are more susceptible to UTIs than men, because unless they are cleaning themselves properly after bowel movements, they can transfer gut bacteria, mainly E. coli, into the genital area. So it's always a good idea to do that.
Okay, so this may not be exactly the point here.
Okay.
If, say, somebody was to incorrectly clean themselves, and then they brought E. coli to the front, didn't shower, and had relations
Yeah.
with mouth to bottom, you know?
Yeah.
Would it be possible for the male to get E. coli like you would from eating food or something?
Yeah.
That's interesting.
Yeah.
That's
Yeah. It's
horrible.
possible. Yeah. Yeah. That does happen. But not just E. coli, all sorts of things.
Wow.
Because it's not, the E. coli is not the only thing that we have in our gut. So yeah, there's that possibility. So again, so you discard that first portion. Again, unless this is a 24-hour collection, you discard that. You avoid the first portion of the urine into the toilet, and then collect your specimen from midstream. And it says to stop when you're about half full. I usually say about 3/4. It's never inappropriate to have a little extra. So, and then cover it for sure, always, and then wash your hands again. Male procedure is very similar in that washing your hands, cleaning the penis well, and nobody ever does this. Nobody ever gives you cleaning supplies when you go to give a urine sample in a doctor's office. So this is an ideal.
For girls?
I don't know about girls. They never did for me.
They do for girls. all
Good
the
for them.
time.
Good.
But yeah,
I have
when my
never,
husband has to go and do a sample, they never give him anything.
yeah,
They don't even tell him, like,
no,
hey, make sure the tip of your penis is clean of debris or
Yeah.
or whatever, and just make sure you're doing it. So tell him to do the stop and, you know, fill it or pee, fill it, stop, drop it. That's it. They never tell him clean it.
yeah,
So I always thought,
and
well, boys, I guess are because they're a straight object.
Um,
It's fine
yeah, and you can't ever assume that.
because they never give them nothing.
No, I have never been told that. You know, but I've, in fact, it might have been when I was a kid. That might have been why I was told I had a kidney infection when I didn't. I wouldn't be surprised. That's funny, but that was a long time ago. Anyway, so again, you know, the issue is just clean. Make sure you're clean. Infants are a little different. What you're seeing here is when we need a urine sample from an infant, because you can't tell a kid to pee. I mean,
Here, catch
it
this
works.
pee.
Yeah.
Well, my 5-year-old could do it. But like, when
Yeah,
he was
at
3,
that age, they should be able
when he
to.
was 3,
Yeah,
it's fine. Like, not right in the bathroom.
yeah, right. Yeah. So usually we can do this at home. We have all sorts of special bags that fit over the vulva or around the penis and scrotum, which have adhesive, and you attach them to the child. At that age, they're not likely to have any body hair there, so it's not going to hurt them. And then put the diaper on them. Okay. Yep. And wait for it to fill. Okay. Isn't that great? Yeah.
That's ingenious.
Yeah, there's a towel there
To make
to
the hole smaller so they don't fall through.
yep, that's
Freaking
clever.
genius.
That's clever, yeah. It's the first time I noticed that. Very good.
Well, my
So-
kid still, he has this little toilet seat, and he's 5, like I said, but he likes his toilet seat because it makes it fit him.
Yeah,
So he still has his little toilet seat and stuff. I'm sitting here thinking like, that looks pretty comfy if I didn't have a toilet seat. That would work
yeah.
out.
Okay, so so we collect this and we attach the bag, put the diaper on the baby, and then check periodically.
But what is the bag like? Because it's in a diaper?
That's why you check them pretty often.
Okay.
You know how often your child usually fills his diaper or her diaper, how often, you know, to an extent, you
Especially
know.
if you're paying attention to the point where they're in a bag.
Yeah,
I think
So you're
the
gonna
parts are
check
gonna be
on it.
pretty attentive.
yeah, and even your 3-year-old can say, well, yeah, I think I peed, mommy.
Right.
You know,
Can
so
you take this off me now?
yeah, so you get this away. So then you drain the bag into a sterile container to take to the lab. Okay, so that's to be careful. Now, do not touch the inside of the cup or the lid. That's so you do not contaminate the sample from your child, because again, we're covered with bacteria and other things. You know, so you can put the container into a bag. And this is always a great idea. If you're going to put a urine sample into your refrigerator, it's probably a good idea to go ahead and put it in a plastic bag so it doesn't totally gross out somebody else. Because you cannot trust that those lids won't leak either.
Because I had to keep filling it and
Right.
turning it in. But when I was pregnant, I was dropping lipids.
Ah,
So they kept having me fill it until I got that under control. And I had to keep putting it in the fridge, and my mom would just be like, "There's pee in the fridge!" Every time she opened the fridge, it was this giant, like, orange bottle.
yeah, they're one of those
And
big
it
orange
looks
ones.
like an orange juice container from like the old folks' home.
Yep.
So I'm sitting here, when I look at it, I'm like, it's juice. Oh no, that's my pee. Don't drink that.
Don't drink that. So with a timed collection, we have the idea is to check throughout the day. We'll secrete different things into our urine. So you might miss something on a random sample. So we're looking at testing the urine throughout that time period. So the idea is to collect it all. This, we're not disposing of the first ounce of each urination. We're
toilet, catch it all.
That's right.
And you pour it in the jug.
Right. And if you happen to put a stool in there, separate it out. Don't worry about it, because they want that urine. If you get a little bit of stool in there, they can deal with that too. Yeah, well, you don't like it, no. Yeah. So, and if you lose a sample, say you spill it, and especially if you're voiding into a pan, then you have to start over with the whole timed thing. So, so it's going to be, 8 or 24 hours usually, and then before 0.0, the time you start, you should start with an empty bladder, so that you go for the amount of time that you're supposed to go, and then at the end of that time, you empty whatever is in your bladder. And you end with it empty, because we want to know everything in that 8 or 24-hour period. Then you need to get it to the lab as quickly as possible. Some usual precautions. Obviously, you're not going to do this when you're menstruating. You're going to have blood all over the place. And
sorry, I just have some visuals.
Yeah, I know. So, make sure that that you time you put a time for each urination. Okay. So some of these are related to the glomerular filtration rate. That's what the creatinine clearance rate test is how well your glomerula are functioning. The urea nitrogen in urine, as opposed to blood urea nitrogen, because the urea appears in urine. Obviously, we call it urea for that. Also, our glucose, because that's going to cycle during the day. The amount of glucose that's going to come out. If you have just consumed, a 16-ounce Coke, you're going to have, not too long afterwards, a lot more sugar in your blood. That's why they want you to even to do glucose testing at least 2 hours after eating, so that we can clear stuff out that's just a spike from food. What we're interested in is something that's long-term. And of course, our electrolytes, and a class of chemicals called catecholamines, which are affected by our diurnal cycle as well, but they're important things as well. And then also the 7-hydroxysteroids, which are hormones that can help us with Cushing's disease, which is a pituitary tumor that overproduces adrenocorticotropic hormone, which can cause a lot of problems. A list here of a few include immune suppression issues, diabetes, depression, several things. Another class was the catecholamines. These are hormones that are made by the adrenals and are stress hormones. And these include the dopamine, norepinephrine, and it can be used to rule out tumors of the adrenals and neuroblastoma cancers. So they're used when there's a reason to want to rule them out. these tests are used. So these are a wide variety of the kinds of tests. 24-hour collections are done. They're an important test because what they're doing is looking at how your kidney is functioning over a period of time, because again, it cycles. Everything cycles. It cycles based on your rate of consuming food and water, and it cycles during the day. Different chemicals are cleared at different times of day and night. These 24 hours include your nighttime samples. And then another reason is that we need some of these is that a random sample is going to miss a lot of them since they're cyclic. So we wanted these 24 hours. It's fairly easy. You know that from doing it yourself. It's a pain in the neck to fill up that orange bottle. Yeah. Yeah, that's right. You want to go back to bed and go to sleep. Yeah.
When I was pregnant, there was more about not being.
Yeah, I bet you did. Yeah, and that's important. That's really why you do it. So so you know how this is done, and what we do is we're looking. One of the major things is looking at the filtration rate, which is measured by creatinine clearance, and we use these light-proof jars like these.
Orange juice.
Yep, orange juice containers.
For old folks home.
Yeah.
Makes no sense.
Nope.
I feel bad for the old folks though. They got those in their fridge, and then they go to the dining room, and they drinking out of them.
Yep. Yep. So our again, we have to be able to give instructions to our patients. So we have to have all of it. We have to emphasize that every drop. Don't go in there and
It's
pee a
okay.
couple ounces. Oh, I got to collect this. You need it all. Okay.
That's
So
one reason why mine took so long. Because the first 2 times, I told him like, well, I first off started off like, please, sorry, my bad. But I clicked the rest of it. Took it in, told him, he doesn't go do it again.
yep. We
And
need
then-
to see
and
every bit.
nighttime,
Yeah,
forgot.
nighttime is, that's the hardest time, I'm sure.
But the first 2 times I did it, I messed it up, and then I had to keep doing it because they couldn't get me to stop dropping the lipids. And they were trying to figure out why.
Yeah,
My kidneys were working,
yeah.
but I was dropping lipids like a lot.
Did they ever figure out why?
No.
And you had a healthy
But
kid?
yep, they put me on vitamin B12,
Yeah,
and also a low sodium diet,
yep.
and kept me from going into preeclampsia. But they never figured out why I was actually dropping the lipids.
Yeah, well, there's a lot of things they don't figure out.
I figured
and-
it's because my body was stressed out.
It could very well be. Stress does weird things to our bodies. And of course, the important thing is that you had a healthy baby. I mean,
Yep,
that's
he's
all we care about.
Brilliant and perfect and nothing wrong.
Excellent. So, and we want to know exactly when you stop or when you start. So, so if you get up in the morning at 6:15 is what they've used here, and that's your first void, write that on the jar, and then do it all day. In between times you're refrigerating it.
You can't leave your house.
Yeah, that's right. Unless you carry it with you in an
I
ice
wasn't
bucket.
taking that.
Yeah. So then, and then the next morning, you want to finish by collecting that first morning urine in the jug. Okay. And note the exact time. So what you want is within 10 minutes of the start time on the first day. So it can be 10 minutes earlier, can be 10 minutes later, but within
But
10
it can't
minutes.
be like you actually voided at 6:15 and you write down 6:40.
Yeah. Not supposed to do that. Okay. So, again, try not to have any feces, but if you do, don't worry about it. So these 24 hours, let's see, just sort of work things out so that you can do this. Okay. So we look at a number of different different metabolites and substances here. We'll get into what these things do. Obviously, arsenic or heavy metals like lead, cadmium, tungsten, few other things that we might get in our urine. We have metabolites of some of the drugs or some of the substances that we know can be produced by a sick adrenal, and then hydroxyproline. So if we're looking at we're looking at some other things for looking at neuroblastoma. We're looking at oxalate and uric acid because that buildup can result in some problems in other parts of the body. So catheters we don't usually use on healthy patients or patients who can urinate on their own. It might be the best and the best way to get a sterile sample or a sample that has no chance of being contaminated outside the body, or to treat local infections within the bladder and cancers, we can inject medications through a catheter. So there are all sorts of tools that we use for collecting these things. You saw the cups the other day. Don't ever trust them not to leak. Some lids are specially designed to be able to transport, to transfer the urine to something else. And then we have some conical tubes, which we will be using on Wednesday to centrifuge samples. Here's a picture, excuse me. And these products, the cup, we have straw devices that we can put down into the cup and suck into an evacuated tube, just like a blood collection tube, samples for various transport and testing. Okay. So we have different kinds of evacuated tubes, which we can send in for different kinds of tests. And an unevacuated tube, we just fill them up. Don't worry about it. Just pour them in. Actually, you should use a transfer pipette.
Like a funnel?
Pardon?
Like a funnel?
No, a pipette is something that looks like a little bitty turkey baster.
Oh, okay.
Okay, so you just basically draw stuff into, because you really don't want pouring a sample that you don't know anything about all over your workspace. Remember from your Med 102 class when you took the OSHA training, we assume anything is potentially contaminated material,
right,
potentially infectious bodily fluid. And urine is one of those. As I say, urine's supposed to be sterile. Blood is supposed to be sterile, but we can't trust that they are. Okay. And one of our best refrigeration, or one of our best preservative functions is the refrigerator. But we also can use various other preservatives. Acetic acid is just vinegar. Hydrochloric acid is also known as muriatic acid. It's pretty nasty. If you don't want to use it, you shouldn't. We can also use formaldehyde or thymol. So Thymol is actually used as an antifungal agent in some lip balms. So it's actually pretty good for cleaning things up. So that's the end of that that lecture, and it's past time for a break. So I'm going to pause. So I'm resuming the recording now. This next PowerPoint, we're only going to go through part of, because we'll do the microscopic analysis part on Wednesday. So there are three major forms or approaches to urinalysis: physical examination, chemical, which we did last week, and then, or some of last week, obviously not the full gamut of tests that can be done, and then microscopic. Physical tests, we look at color, turbidity, meaning how cloudy it is, volume, odor, and we can measure the specific gravity. Normal urine is straw colored. Normal urine is what we're talking about when people drink adequate amounts of water, and their diet is reasonable, and by reasonable I mean that's a wide range, and yeah, and it varies from person to person, and also whether you're healthy or not. So the normal color ranges can be from basically straw colored to dark yellow brown. Darker in the morning for the most part because you've been concentrating stuff overnight, and during the day as you build up and dispose of urine, it'll be different colors.
What if your teeth is the same color all day, super dark all day long?
Then you're probably not drinking enough water. That would be one thing. Another is that you probably need to have some sort of test to see what is causing the darkness. Okay. So there are drugs that can change your color, and foods will do this. We all know about asparagus urine, pardon? And blue and all sorts of colors, red. Yep.
Bright orange?
Yep. They will, and some antibiotics are bad about that, but other colors are not so nice. Red or pink usually means blood, but also if you eat a lot of beets, things that are red, you're going to get that. And some anemias will do that. Other colors can be from food or can be from breakdown products of other parts of the body. But it really, the color itself only tells you if it's not a normal sort of straw yellow, that you should be looking at something else. Yeah, it stinks. Yeah, so that's part of the odor. Okay. So people produce usually 1 to 2 liters, or about 1 to 2 quarts of urine a day. More than that, we call it polyuria. Less than 400 milliliters a day, we call it oliguria, meaning partial. And then anuria, less than 100 mils a day, you're not peeing. That's a very bad sign, actually. Random volume samples generally run from about that 10 milliliters we want, can be as high as 150. Depends on your bladder size, how recently you've urinated. That the volume of a random sample is not terribly important. Volume is really important when we look at these timed samples. Easiest way to measure is to use measuring equipment from a chemistry lab. Odors. Most of the time urine has a pretty mild odor. And most odors don't indicate a health problem. There are some medications and other things that can affect it, and foods. Asparagus, there is an amino acid, asparagine, and that's what you're smelling. But not everybody does that. Not everyone has that reaction.
And the
So.
kids are absorbing more nutrition.
No, it has nothing to do with that. It has to do with how we metabolize proteins. So if you don't release the asparagine, which some protein digestive processes won't, you're not going to get that odor. But it can range from sweet to foul. Color often has nothing to do with it. You can have urine that's black because it's so full of things like bilirubin if you've got gallbladder problems. That has no odor, basically. Sweet, we're worried about sugar-based disease, diabetes mainly. As I said in class the other day, the word diabetes actually comes from sweet, a Greek word referring to the sweet smell of urine. A foul smell often is related to an infection of some sort. This musty odor, there is a metabolic disease known as PKU or phenylketonuria that we look at in children. We test children for when they're born to see if they're phenylketonurics. That changes the way they can eat. Specific gravity, yes. It's not there? Oh dear. All right, so this silly thing turned itself off again. Thank you. It's going to be in the recording, but this is kind of oddball here. Don't know why it's doing this. Okay. So now it should be. Okay, yeah, there it is. All right, so specific gravity basically measures how much stuff besides water. Specific gravity is a comparison with the weight of a fixed volume of pure water versus your sample. So for instance, pure water at 4 degrees centigrade, a milliliter weighs a gram. If you put anything in that water, it will change the weight. By anything, I mean you can mix in other liquids and make it lighter. You can mix in solids and make it heavier. If you mix in alcohol, it actually gets heavier because it actually shrinks the volume a little bit. So the specific gravity of alcohol-containing liquids is higher than 1. And 1 is the reference. So
if you have something that
well, the alcohol comes out in the urine because alcohol just gets cleared very well by the kidneys. Alcohol is a poison, and the body gets rid of poisons as fast as it can. So and we get rid of it several ways. The balloon test for alcohol, if they stop you on the street, that's because we exhale it because it's leaving our bloodstream. Because it enters the bloodstream pretty much unchanged. And then it starts tearing up everything else after that. And then we'll also excrete it through the kidneys, and we can measure that. So the normal specific gravity of urine is somewhere between 1.003, because there's always something in it, to 1.035. That's why we have the test levels in that chemical strip at those numbers. So that's normal. And the more junk you have in there, the more the higher the specific gravity. So you'd expect specific gravity in the morning to be higher than at midday when you've been drinking water. Okay. So one of the things that that can cause low specific gravity is the diabetes you're going to be looking at, glomerular disease and kidney disease where you can't reabsorb water from the urine, and of course renal failure. High specific gravity, dehydration. We talked about that. Diarrhea and vomiting. This is when you're really getting a lot of water out of your system and not able to replace it. Heart failure leads to dehydration. Shock, lowering of blood volume, and then other diseases as well. So there's several methods. The most, this particular method is not something we use. This hydrometer, they're cheap. They're not very accurate, and they're very temperature dependent. Mostly we'll use this handheld device called a refractometer that actually measures the refraction of light through a sample, and the greater the refraction, the greater the specific gravity. It's a pretty easy instrument to use, and it's a handheld instrument. It's about this long, about 8 inches long. So it's fairly easy. It's fairly readily available. It is really accurate. And then there's a dipstick test that is the most common way. We looked at specific gravity as a chemical test the other day, or last week. And it is the most common method because it's actually the cheapest. The refractometers are, can't remember. I bought a couple of them down for the lab in Denver, and I think they were about, I think I got them on sale for about $50 each. You can buy an awful lot of strips for $50.
Oh my God.
Okay.
So what would a urologist probably have that in their office?
They would be looking for the real accuracy of that. And they would follow up. They'll use the dipsticks first, just like everybody else. And what they'll do is follow up. If it comes on the dipstick out of range, they'll probably do a refractometer. Okay. So pH is it's a measure of acidity. pH less than 7 is considered acidic, greater than 7 is basic or alkaline. pH normally runs anywhere from 5 to 8. And again, this is dependent on when you collect it. Now, blood pH is 7.35, and it should stay at pretty much at that level at all times. Okay, but it's really dependent on diet, metabolism, and other things. Proteins, we can detect these things chemically. That was one of the tests we did. Most of the time, we're measuring albumin. But we also measure protein, general protein. Albumin is also indicative of some other protein problems in the kidney. There can be false negatives. So protein in the kidney can be caused by this laundry list of conditions. And again, what these chemical tests do is prompt us to do more testing. The proteins, the type of protein that's in urine has to be determined by a test that's not our standard CLIA-Wave test. They can tell us a lot about what's going on with the kidneys. And it's not unusual to see proteinuria in UTIs. The glucose test that we have is pretty specific for glucose. It will not detect other sugars. And there are plenty of sugars. We see them all the time: galactose, lactose, fructose. These are everywhere. So if you're drinking a lot of high fructose corn syrup containing drinks, and that sugar is being cleared through the nephron, this won't pick it up. So you need other tests to really do that. And the urine there really does need to be at room temperature. Ketones are a breakdown product or an oxidation product of sugars, and fats. Fatty acids get broken down and then get modified to ketones. the glycerol carrying body can be oxidized to ketones. So if we're, and we see ketones, we can measure ketones in blood serum, and then that alerts us to look at the urine and vice versa. Hello, Katie.
They've kind of been working on their own and series we've been watching them. I have given permission to leave at 9.
Okay,
Because
so
the instructor is not here, and so
they're going to leave at 9. That's great.
it's, yeah, it's fine.
Okay,
All right.
super.
Thanks.
See you, well, Wednesday. See you Wednesday. Thanks, you too. I've locked up the med labs. Yeah, I was leaving that, so a more comfortable chair. Yeah, I don't blame you. So there are ketones can arise from a number of different sources in the urine, and the third choice, starvation, fasting, and dieting. This actually is something that happens in the so-called ketogenic diet. We generate these ketones in the urine. So there can be other causes. Pardon? Short term. You should never, if you go on a ketogenic diet, it should not be a long-term diet, because you're generating things here that can have long-term effects. So we do these. We do part of the ketone test tells us how well controlled diabetes is, these are useful in the endocrinologist's office as well, monitoring what's going on. Blood can come from a lot of problems, including trauma, including pardon?
Working out.
Yeah. And just a lot of different things can happen, and UTIs can give you blood. Obviously, menstruation will but the blood's not coming from the urinary system. Then obviously, it's just mixing with the urine. Cystitis, which is an inflammation of the bladder. Cyst means sac, so the bladder is a sac. So cystitis is just basically an inflammation of the bladder that can then make the bladder bleed and other things. So blood levels, we're going to look at blood cells and also hemoglobin from the blood. There are lots of, but hemoglobin isn't common, at least not in this country. Except under certain conditions. So, pardon? Hemoglobin is the oxygen-carrying molecule in red blood cells. And when those, we don't like to see that in blood, because that means the red blood cells are getting destroyed. It can indicate certain kinds of cancers, but it's not generally a cancer marker. It can be related in there. So it's we can have lots of if we can't we see red blood cells in microscopic exam. We'll talk about that on Wednesday. But we seldom see hemoglobin issues. Myoglobin is a protein found in muscle, and muscle injury and shock and actually multiple sclerosis and certain kinds of poisons can result in seeing that particular one. Bilirubin is excreted, formed in the spleen when hemoglobin is broken down. It's how we get rid of broken down hemoglobin. It goes into bilirubin, and there can be when it's in blood, you're probably going to see it in the urine because that indicates that you've got some issues with liver or whatever. Bilirubin is used as a measure of how well our liver and gallbladder are functioning. If there's a high amount, we don't like to see a high amount. That means that we're producing way too much. Okay. Because it's normal. I mean, we secrete it all the time. Then urobilinogen is bilirubin breakdown. This comes out of the intestine, but is excreted by the kidneys. Small amounts fine. We're going to see that fairly regularly. Larger amounts indicate problems with the destruction of red blood cells, or liver or intestinal problems. So this table here just tells you what kinds of conditions. You can access this PowerPoint through your shell and take a closer look at it. We look at leukocytes, white blood cells, by measuring an enzyme called leukocyte esterase. We don't, it's quite normal to see some the same as red blood cells. Is quite normal to see some in the urine because it's part of the way we get rid of old and dying cells. They do die. Higher numbers, however, we're really worried about UTI or other infections and other potential problems. And again, this is another one we confirm with a microscopic exam. Nitrates are bacterial in origin, indicating a UTI. We don't like them because we see them in gram-negative bacilli, which are particularly nasty bacteria. And again, we can confirm that with a microscopic exam. Many, there are some that are. Bacilli are the rod-shaped bacteria. They cause things like gonorrhea, and a lot of us carry bacilli that are perfectly benign, and so it really it depends on the level and how much. Okay. So we actually do these things. So we're going to stop here because the microscopic exam will be the subject where we start up again on Wednesday, because we're really looking at structures that can be visualized as opposed to chemicals, which cannot. Things that we can actually see in the field of a microscope. Okay, so we'll go through microscope use and then and sample prep for microscopic exam. So questions? Okay, so let's go back to the let's go back. Where am I? Let's see, I want to escape. I want to get rid of that. Screen sharing is paused. Now I want to go to where, what's going on here, folks. Okay, now we want to go to that. Why isn't that showing up? Now we'll go to a share. And here we go. All right. So go ahead and get started on the diabetes assignment. We have a bit over an hour left tonight here at school. Those of you watching this, send me an email with the word Swansea, SWANSEA, in the text of the email or the title to let me know that you've watched this, and get started on some of these other parts. The discussion, read that, take a look and do some research on the medical assistant's role in your analysis. Start work on this presentation and start doing the videos and the second set of quizzes that we have, the assessments we have for this week. And I will see you guys hopefully on Wednesday.